A sarcomere is composed of thick and red and thin and blue filaments. The thick filaments are made of many myosin. Myosin has a tail and two heads, which will move back and forth, providing the power stroke for muscle contraction. Myosin is in its low energy conformation when the cross bridge is in this position. The tail of myosin has a hinge which allows vertical movement so that the cross bridge can bind to actin.
The thin filaments are made of three protein molecules: actin, trypomyosin, and troponin. Actin is composed of a double strand of actin subunits, each of which contain myosin binding sites. Trypomyosin twists around the actin. When sarcomere is not shortening, the position of the trypomyosin covers the binding site on the actin subunit. Troponin functions to move the trypomyosin aside, exposing the myosin binding site.
During contraction of a muscle, calcium rushes into the cytosol and binds to troponin.
This moves tropomyosin out of the way and uncovers binding site for myosin on the actin filament. Also note, myosin must be activated before a cross-bridge cycle can begin.
ATP binds to myosin head and is hydrolyzed to ADP and phosphate. Energy is released into the myosin head, causing it to ascend into a high energy position. The activated myosin head binds to actin site, forming a cross bridge. ADP and phosphate are released, and the activated myosin head pivots, sliding the thin filament towards the center of the sarcomere. Pivots, sliding the thin filaments towards the center of the sarcomere. The myosin head flexes into a lower energy position while attached to the thin filament. This is called a power stroke. The myosin head released from the active site of thin filaments when a new ATP attaches to the myosin head. The energy release reactivates the myosin head, returning it back. As long as the binding sites on actin remain exposed, the cross-bridge cycle will repeat.
And as long as the cycle repeats, the thin filaments are pulled towards each other and the sarcomere will shorten. Cross-bridge cycling ends when calcium ions are actively transported back into the sarcoplasmic reticulum.
and thin yellow.
What?
The thick filaments are made of many myosin.
Hold on, you forgot to say blue and red. Blue.
And red, and thin blue and yellow filaments. No. Myosin has a tail and two heads, which will move back and forth, providing the power stroke for muscle contraction.
Let me point to the two heads.
Oh, okay.
The thin filaments are made of 3 protein molecules: actin, troponin, each of which contain myosin binding site. Tropomyosin twists around the actin. When sarcomere is not shortening, the position of the tropomyosin covers the binding site of the actin subunits. Troponin functions to move the What?
The energy released activates the myosin head, turning it to the cock position.
What?
You have to pull it. The energy release reactivates the myosin head, returning it to the cock position. Energy.
It's not even funny, man. As long as the cycle repeats, the thin filaments are pulled towards each other and the sarcomere will shorten. Cross-bridge cycling ends when calcium ions are actively transported back into sarcoplasmic reticulum.